Good evening. You are answering test today. What I want to call. Oh, you were there in non-breaks. So yesterday I think we discussed a lot of things in general. Okay, like very little about the chapter and here and there many things we were discussing. Okay, fine. Good now. So today, as the continuation of the yesterday's contents. First, we just this is about the mechanism of in yesterday's class, we discussed about the definition for reproduction and the significance of reproduction. Okay, so can you tell me the definition for reproduction? Can you define reproduction? It is the biological process by which progeny or progenitors. Okay. Or will learn to their very good. So that's about the definition for reproduction. Fine. Significance of reproduction. So produce more and circulate. Okay, just one thing, one word. What is it? Life form. Okay. Dash of life form. The first letter.
It's a continuous form of continuous form.
Of, very good, very good. Continuity of life forms. Very good. So that's the main significance of reproduction. Okay, fine. Keep that in mind. Continuity of life form. Very, very important thing. Okay, fine. Next. Today we will discuss this topic, the mechanism of reproduction. What actually occurs in reproduction? Both in sexual as well as asexual. So this is the main mechanism actually. Like in one word, you want to say mechanism. That is main mechanism by DNA copying mechanism or DNA copying mechanism or DNA copying process mechanism, or you can say process. Okay. One liner about the mechanism of reproduction. What actually occurs in reproduction? What process is the main mechanism involved in reproduction? DNA copying process. One liner. Okay. Significance of reproduction. What is the one liner? Continuity of life form. Okay, or continuity of species. Continuity of life form or continuity of species. That's the one liner for what? Significance of reproduction. Mechanism of reproduction. What is the one liner? What's the one liner? DNA copy mechanism, or it is also called what? DNA copy process. Okay, that's the one liner for mechanism. See now, let's discuss this DNA copy mechanism in reproduction. With an example of a mode of reproduction. What is the simple mode of reproduction? It is binary fission, which is the simplest mode of reproduction. It is what? Binary fission. Okay, simplest mode of reproduction is what? Binary fission, which is the simplest form of living organism. Which is the simplest form of living organism? Bacteria. Bacteria is the simplest form, unicellular, small system, microscopic, prokaryotic organism. Okay, bacteria are unicellular, microscopic, prokaryotic organism, the simplest form of living organism, and they reproduces by what mechanism? Is the unicellular simplest microscopic living organism, bacteria, reproduces by what mechanism? Binary fission. Okay, which is the most simplest form of reproduction process? Binary fission. Okay, how? In this simplest form of reproduction, binary process, binary fission, DNA copying mechanisms is involved. That's what we are going to discuss now. Okay, let me start explaining the DNA copying process by example of a simplest cell division or reproduction process, binary fission. How can we define binary fission? One parent organism split into two identical individuals. Two identical, you can say, offsprings. Okay. One parent organism split into actually two equal off. Okay, one organism split into two equal off or two identical organisms. One parent, two organisms. That is called what? Binary fission. For example, the simplest diagrammatic representation of binary fission is this is one organism called a bacteria. Okay, and this bacteria can produce two different means identical organisms. Okay, one parent organism produces two daughter cells. This is binary fission. One divides into two, and two daughter cells are formed in binary fission. Pi means two, fission means division. What's called fission? Fission means what? Division. Okay, fine. Now just assume, say, look into the diagram. If this is the parent organism which produces two equal of, means actually what should, if it produces two equal of, it should be like this. Yes or no? If a parent organism divides into two equal of, it should be like this. How? This and this are identical. It would be identical. Yesterday we used the term for identical. What is it? Clone, very good. Clone means identical. Now, which is correct? Here, we do an option. And here are two daughter organisms. Equal organisms. See, look at that. This is the correct method, or this is the correct method. Which is the correct method? You get my question first. One current organism, two daughter cells. Equal of binary fission means 1 into 2. You have one bread there. I'm asking you to share. Off of only intake. This is the picture here. Okay, in binary fission, this address or you have one bread. I'm asking you to divide. You get one bread, full bread. You get one full bread. What is possible? This one possible or this one possible? Huh? Actually, logically speaking, if it is a bread, I'm asking you to share. You get off of the bread. Okay? But in binary fission, actually the daughter cells are what? I told you one term. Daughter cells are what? Actually, two identical are thing. Identical to the parents, they have clones actually. Then it should be size, if it is this much, same here and same here. How it occurs?
Then what happens to the parent? What happens to?
Yeah, parent is no more existing. That itself is actually producing two. Can you understand? So,
size.
uh,
Of the size is producing.
no, completely a kind of regeneration. Means completely it is new now. The old undergoes some changes and then it is new now. It is newly born. One newly born into two. And then that is newly born into two. Okay, that's why they are called what? Inverted. In our reproduction process, there is existence of what? Tata Patti, Appa Amma, 20%. So can you see the differences in age, bodily characters? Yes or no? In sexual reproduction, you can see grandparents are very old, parents are some 50s or 40s, 20%, 10, 20, whatever. You can see the difference. But here, the speciality of this mode of reproduction is when an organism reproduces by binary division, the equal division, here there is no existence of the old cell. Actually, both are new now. New only. Okay, no existence of old cell. New one developed into two new. And then each one developed into 230. That's a very, very important process in binary division. See, then this to occur in between what? Just like that, a parent cell cannot divide into two people. So for its division, in between the loss setting process must occur. Can you understand? So that process will give you better explanation for this process. Okay, now how this process, this is not going to occur, only this size going to be there. How bacteria reproduce like that. That's what we are going to discuss now based on this diagram. Okay, can you see the diagram here? Actually, what organisms they are using? Amoeba. Okay, for binary division, they are using amoeba. So, I'm going to use the same diagram actually for your better understanding. Okay, how amoeba reproduces by binary division. What is amoeba? You can see even lot of what they are basically are called. Pseudopodia. Okay, this is membrane, cell membrane. Okay, and then cytoplasm. plasma, and then this is nucleus, and the extension of the membrane, that is called what? Pseudopods. Okay, pseudopodia, very defining characteristics of amoeba. The shape is irregular. Okay, and also amoeba possesses this shape, then it is called what? Amoeboid shape. Okay, even can you give me an example of a cell in our body which can have this shape, amoeboid shape? Same amoeboid shape. We have a cell in our blood which can. No, or this is this shaped cells. Right? So, white blood cells and especially WBCs, neutrophils. Have you heard about this cell? Neutrophil, which is having amoeboid shape. Okay, fine. So, this is amoeba. Now, how this amoeba reproduces two amoeba. Okay, same size like you should have a amoeba. How many? Maybe like two cells you should have. Maybe my size is different, but it is the same two cells you should have in a. Okay, two amoeba. One amoeba should produce two daughter cells. Okay, in between, what is going to occur? That's what we are going to discuss now. Okay, the first process in binary division of amoeba, what should occur? Say something called what? We can go down A, and here I think will be elongation. Okay, elongation. What is called elongation? You know extension, simple term, extension. What is called extension? See, you have an upper cut. What you can do? Like this, you can extend its size, as well as like this, you can extend. So that is called what? See, longitudinal and transverse elongation, all size. So the whole amoeba increases its cytoplasmic content. Can you understand? So like this, irregular shape. So the length as well as the width, both. Can you see the extension of the amoeba? That is the first system. What is the first system in binary division? The parent organism before division extends its size and content inside it. Okay, it has to divide into two. So, for that division, what are not required, so it accumulates all the nutrients. Okay, that's the first thing. Lot of water, lot of nutrients, and everything it just accumulates in the cytoplasm. That's why it gets extended. That's the first thing. Okay. What is the second process? Second step in that extension should be followed by what? Division of nucleus, especially before division of nucleus. The second process which is going to occur is daughter cells. They are actually called daughter cells. Parents and daughter cells. The progeries are called daughter cells. You are asking why they are not sons? Why they are only called daughter cells? Okay, because can you give birth to your egg one? I don't know that. Yeah, daughter cells are since because of the ability of reproduction, like common lay one term, what do you call it? Daughter cells. We do not call it as son cell. Okay. Now, what is the second process? elongation, then DNA replication. This is the main process. What is it? DNA replication. What is it actually? DNA copying process. DNA replication is also called here what? DNA copying process. So copy, photocopy is the thing. I mean, when to a shop, to take photocopy. What will they do? You give one sheet. You need 2 copies. This is 3 copies. So that is copying process. Similarly, like that here, DNA is going to produce its own copy. Okay, that's the third step. Second step basically, DNA copying process. So this DNA which is present here, okay, they may have some DNA. Actually, inside the nucleus, this is nucleus, that DNA, one DNA is going to be here, for example, for your better understanding, it has one DNA. Okay, the amoeba has one DNA. Now, this one DNA, it is actually nucleus is elongated and one DNA converted into what? 2 DNA. This process, this is again the amoeba. Okay, amoeba. One DNA divides into 2. That process is called what? DNA replication. Replica. What is called replica? Identical copy. Replica. Exact copy. Replica means what? Replica. Exact copy is called a replica. So that process is called what? Replication. Making exact copy. That is called what? Replication. See, in all modes of reproduction, the organism have to reproduce or now. What is the necessary process to be noted? Yeah, the DNA should be replicated. The DNA should be copied. One DNA should be made into 2. How many chromosomes you have? What that is in our cell? It is amoeba. 1 DNA, 2 DNA. Okay. How many chromosomes we have actually? 23 pairs. Good. Okay. 23 DNA. This copies chromosomes into 2 1. 46. Same as zygote. Here division, but in our body it is fusion. Fission and fusion. What is the difference between fission and fusion?
Fission dividing.
Division. Fusion is union. How this replication is the sense what they occurs. Sperm cell brings 23 chromosomes. Egg cell brings 23 chromosomes. Now it is 46. DNA is multiplied. 1 DNA, now 2 DNA. 23 chromosomes. So now 46 chromosomes. But here it is one parent only produces 2 cells. So what it has to do? One DNA be duplicated. That is a very, very essential process. That is DNA copying mechanism. Can you understand? DNA copying mechanism. That's why if they ask one question, what is the main mechanism involved in reproduction? It may be sexual reproduction or it may be asexual reproduction. What is the main mechanisms involved in it? DNA copying mechanism. Also called what? DNA replication. Can you understand? Replication means production of exact copies. That's the main very important thing. Okay, fine. Now it should be followed by what? What is the third process? DNA replication. So now listen. Karyokinesis. What is the next process? Karyokinesis. Carry on represents what? Yeah, karyokinesis. Carry on means nucleus. Carry on represents what? Nucleus. Kinesis again division. Okay, kinesis means division. Upon karyokinesis means what? Nuclear division process. Is it clear? What occurs in what are the steps? Now, first, elongation, enlargement. Second, replication, DNA replication, duplication of the DNA. Third, nuclear division, karyokinesis. Okay. Third, fourth step means what? What is the fourth step? Or B, you can say the cytoplasm has to divide, right? Next, see like this, the cytoplasm has to divide. It has to divide. Okay, so nucleus also division. Actually, here one nucleus like this nuclear division and then cytoplasm. Cytokinesis. Okay, division of the cytoplasm. Okay, division of cytoplasm or cell division, you can say. So that produces what? Two daughter cells. Can you understand now? Look into the image now. Okay, so listen, look into your image now. Okay, the first is amoeba. Okay, next, can you see the size is elongated? Okay, and then can you see nuclear division, and the nucleus also starts dividing. Can you see the extension in the nucleus, division of the nucleus, and then cytokinesis starts, and finally one parent organism produces two daughter cells. Okay, for this to occur, what is the main essential process which is occurring inside? DNA duplication and DNA division. Can you understand? Inside the nucleus, DNA replicates and then nucleus divides, and then the cytoplasm divides. Okay, so now coming to the question, what is the question? Mechanism of reproduction. What is the main mechanism involved in reproduction? DNA copying mechanism. Okay, and the steps are given here. In a book, you can very well understand the thing. Now, this is the processes: elongation, DNA replication, karyokinesis, cytokinesis. Finally, two identical offsprings are formed. Is it clear? This process is called what? Binary division. Okay, just we use this process to explain what process? DNA copying mechanism. Okay. Now the progeny or this is a method of asexual reproduction. The progenies are identical to the parent cell. Clones. The progenies are called what? Clones. Okay. See, now you saw fission process, which is in the asexual reproduction, DNA copying mechanism in asexual reproduction. Now the same thing we are going to discuss related to sexual reproduction, especially humans. Okay. Now, you know what occurs in sexual reproduction? Sperm cell, and then egg cell. In sperm cell, nucleus, and egg cell we have nucleus. How many chromosomes? 23. Here, 23. Okay, they are called haploid cells. Why they are called haploid cells? One end. Chromosome number is 1 end. That's a haploid, single set of chromosome. Haploid means what? Single set of chromosome. That's haploid condition. In fertilization, what occurs? This nucleus is 23 chromosomes, and this nucleus 23 chromosomes. But the sperm cell fertilizes egg cell. What comes? What is this called? With after the fusion, 46 chromosomes. This is called what? Yeah, processes fertilization. Okay, fertilization. So, sperm cell fertilizes egg cells, both haploid cells form a diploid. This cell is called what? Diploid. Diploid, this dye represents 2. Ploidy represents chromosome number. Ploidy means chromosome number. Okay, so 2 set of chromosomes as zygote is formed. Okay, now paternal genes. From here, what comes? Paternal. From here, what comes? maternal genes. Paternal and maternal genes. Now, actually, like because of that only, you get paternal as well as maternal characters. So here, what is achieved as we discussed yesterday? Genetic recombination. Form haploid fuses and forms fusion and form diploid cell. So here what is there? Genetic recombination. Because of the fusion, two different set of genes are united here. So this is called what? Genetic recombination. So here it is Appa and here it is Amma. Sperm cell carries Appa character, egg cell carries Amma character. Because of like 50% of Appa character and 50% of Amma character, but in the progeny, if the zygote develops into an individual, if the zygote develops into an individual organisms, do you see 50% of Appa and 50% of Amma? Maybe. We cannot say maybe. Or 80% of Appa, 20% of Amma. Or sometimes 90% of Appa, 10% of Amma. Or sometimes 90% of Appa, 10% of Amma. Anything. See, this combination may be off. You cannot predict. Okay. How the gene combines, how it expresses. Every gene has its own character. For example, the father has dominant gene. Only father character. Mother has dominant gene. Mother character. Can you understand? So here, dominant in the sense, if a gene is there, we have for every character. Okay, we have two genes in a diploid organism. We have two genes. For one gene comes from Appa, one gene comes from Appa. Naturally, in these two genes, one gene is dominant, means that is powerful. Always it expresses. The other one is recessive. For example, these are the two fingers. One is dominant gene, one is recessive gene. Always the dominant gene expresses. Only in the absence of dominant gene, two recessive genes are there, the recessive gene can express. Weak genes, strong genes, weak genes. Strong genes always expresses. Weak gene, only in the absence of strong gene, it expresses. We will be discussing about that in genetics part also, but for every character we have dominant gene and recessive gene for most of the characters. Okay. So any gene can express. That's why we are combination of what Appa and Amma. Anyway, what is achieved in sexual reproduction? Genetic recombination. Genetic recombination results in what? Sexual recombination. Reproduction results in what? Variation. Okay. Now, in asexual reproduction, progenies are identical to the parents. Offspring are identical to the parents. In sexual reproduction, progeny slightly vary from the parents. Okay. This variation. Okay. Okay, because of genetic recombination. Variation because of what? Genetic recombination. See the percentage of variation. Okay, how can we define variation? The degree in which progeny differs from the parents. Okay, simply how can we define variation? The percentage, or you can say the degree in which progeny differs from parent. Okay. The degree by which the progeny differs from the parents. See, little differences you can spot between yourself and your parents. Okay, that is variation. Can you understand? In asexual reproduction, no variation. In sexual reproduction, little variation. Can you understand? Little variation is there. Now, What is the advantage of sexual reproduction? Clothes are produced. Okay, here advantage of variation or sexual reproduction. In the solar law, what can you say? Note this phrase. Accumulation of variation results in speciation. Okay, accumulation of variation results in speciation. Repeat with me. Accumulation of variation results in speciation. Okay, I come here. Accumulation of speciation. For example, see, like in evolution, I think we discussed from monkeys only humans evolved. Okay, from monkeys only humans evolved. See the accumulation of variation. See, 98%. What is the difference between our DNA and monkey's DNA? What is the difference between our DNA and monkey's DNA? Less than 2%. Our DNA, monkey DNA would improve different less than 2%. Okay, and before going to that, enrolled DNA, how much percentage variation is there between my DNA and your DNA? See, you look different from me, right? Yes or no? In what way my DNA differs from your DNA? How do you tell me? My DNA, your DNA.
Your DNA is very different from my DNA.
Very different from my DNA. Percentage, sir, 1% difference, sir. 2% difference, sir. 10% difference, sir. 50% difference, sir. Yes.
If as you said yesterday,
Mm.
if as per the natural decision, the as per the condition, the monkey got into one woman. So if
Monkey?
the
Yeah, from monkeys humans evolved. Yes.
so there would be a one woman.
Mm.
So from God one woman, the entire world will be.
See is there.
So
Good.
how just one monkey?
It is no. See, no, not so. Not just one humans. Not just one humans actually. Slowly. That's what to explain that only. Accumulation of variation, not monkey to human actually. So how it occurred? This 2% difference between humans and monkeys actually. Initially, the monkey produced another monkey which is having little difference. How much difference? Okay, what is the percentage of difference between the monkey and this monkey? This much only. Maybe for this to occur, 10,000 years. This variation to get permanent, continuous career, how many years it took? Example, 10,000 years. And then after 10,000 years, okay, after 10,000 years, finally, maybe here, less than 2, you can say 2%. Okay, this took how many years? 1.5 million years. 1.5 million years. Can you understand? So, from monkeys to human, you are not one organism here produced. Many, all the species, not just one, all the species carried that accumulation of variation. Variation is accumulated. Species to species to species to species to species to species. And finally, they are not in between So many species that bond ability. Is it clear? Not just one. Actually, it represents a species. Species means what? All humans are one species. Okay, I mean it a species, not one organism. Human, not one human. Human species. Okay, fine. So, accumulation of variation results in speciation. Can this variation naturally occur in asexual reproduction? As of now, what we are discussing, clones are produced in asexual reproduction. In higher class, we study different mechanisms, basically. But now, fine, asexual reproduction, clones. Only sexual reproduction, you have variation. Okay, because of this variation only, new species are formed. Now, coming to the questions which I asked. Okay, percentage of difference between your DNA and my DNA. Why we are all called same species?
Because we are all
Same genetic lineage. Because my DNA matches with your DNA 99.9%. What is the answer? Hari. My DNA matches with Hari DNA with how much percentage? You agree with me? You wonder about this or what? Sir, Thomas sir, no, நீங்க எப்படி இருக்கீங்க? Hari எப்படி ஸ்மார்ட்டா இருக்கா? எப்படி சார் நீங்க Hari கூட போய் உங்களை கம்பேர் பண்ணலாம்? Smart இல்லை. But actually what? 99.9% of all human DNA is smart. Same. அப்படியே DNA உங்க DNA மாறிடுச்சுன்னா என்ன ஆயிடும்? என்ன அர்த்தம்? எப்போ இந்த டிஃபரன்சஸ் கம்மியா இருக்கும்? 23,000 years are 99.9% of the stability. Okay, sexual reproduction ஓட இன்னொரு stability என்ன? இந்த DNA ஓட copying mechanism. What is that? இப்ப நம்ம என்ன mechanism பார்த்துட்டு இருக்கோம்? What mechanism we are discussing? DNA copying mechanism. Even though in sexual reproduction variation comes, what is the main aspect? Why human being can able to produce same kind of human being? In the copying process, what is not occurring? பெருசா DNA change பண்ணிக்கிறது இல்ல. Just color pencils. Colors, for example, you written on your 1000 pencils கொடுத்தா 1000 pencils. Okay, with a different color. Okay, I'm asking you take 200 each. Okay, all of you can get 200. Your combination is different. Your combination is different. Your combination is different. Your combination is different. But actually, what? The gene pool, which is called what? Gene pool. Only from the 1000 genes, you are getting 200, 200, 200. That's it. Okay, we have many genes for the characters in human population. Only that is there different. I have a short gene. He is having a height gene. But gene is what? Height gene. Color gene. I have one gene and she is having another gene. But color gene is similar. So, set of genes. Maybe the combination vary, but the kind of genes are the same. Okay, maybe in your higher class you understand this better. So, what you should know?
So does that mean, like, for example, if I'm Hindu, and the same genes which Einstein had?
Yes. Variation, little variation. See, listen. See, for example, listen. Something called one concept is there. What is your blood group? See, there are 3 genes: IA, IB, IO, or you can say small. These are dominant gene. This is recessive gene. What's your blood group? Correct. If O positive means, listen, your genetic combination is IO. Both are recessive genes. So no A is expressed, B is expressed. And you have positive, recessive positive antigen. That's why you're O positive. Okay? Many people can be like that. Now, my blood group is A positive. What is my combination? IA from Amma, IO from Amma, or IA from Amma, IA from Amma. Same. What is my blood group now? A only. Again, positive gene I got, so A positive. That's different. Positive. Is it clear? What is your blood group?
Because
B positive. So you get this combination from Appa IB, Amma IB, or from Appa IB, Amma IB. In your blood group is B positive. So all are having different people. For example, what's your blood group? You just said A1 fine. That's a little difference only. You just assume from today blood group is AB positive. How you get AB positive? A from Appa, IA from Appa, IB from Appa. So it's AB positive.
So the one here, man.
That's it. That is in A antigen. A little protein chain varies, but that is not going to make any difference. A1 positive, just one protein chain differs. So that is not going to make any difference actually. I can give blood to him. He can give blood to me, whatever. Okay, so these many, listen. These many blood groups, how many blood groups at least here we saw? 4. Here the variations are like positive or negative. So all these are determined by how many genes? Just 3 genes. IA, IB, IO. 3 genes only gave 8 different blood groups, at least 4 different blood groups. That is why we differ in what? Getting these 2 genes. For example, out of 3, any 2 we can get. Okay? So the set of genes are same, but the combination, even though for one character you have many genes for one character, the color, look at the color, not all are of same color, the height, all of same height, because many genes determines your height and skin color. Many genes determines your height and skin color, but I have only 2 genes, few genes only. Is it clear? So getting the particular genes is different. Otherwise, same genes are existing in the population. Whole population, human population, that group is determined by just 3 genes.
so but something to get the lab.
That is actually here in this, I told you dominant and recessive. Okay, A is dominant, IA gene is dominant, IB gene is dominant, IO is recessive. Okay, IO is recessive. They get I go from Appa, I go from Amma. Similarly, like that, there is one more gene called rhesus antigen. What is it? Rhesus antigen. If this antigen, if this gene is present, you get positive. If it is absent, you get negative. Again, the positive gene is dominant. So most of us are 98% people are dominant gene only, having not dominant blood group only. Out of 100, just 2 to 6 people, maximum 8. Out of 100, you count that. Very few people only get negative blood groups. Okay, that is recessive gene. In a rare blood group, rhesus antigen in the red blood cells. See, look at that. This is the red blood cell. In the red blood cell, if A antigen is present, it is blood group A. If B is present, it is B. If AB is present, it is AB. If rhesus antigen is present, it is AB positive like that. So, in the red blood cell surface, you do not have rhesus antigen. You do not have B antigen. You do not have A antigen. No antigens are present in the red blood cell membrane. Very rarely in plants. That is called what? A means O negative. Okay, we saw that movie. Okay, rare blood group actually in the sense, take it is called actually common blood group. Okay, so no antigens are present. Okay, antibody or it's nil, or it's nil means the antigen means not present in the red blood cell samples. Very rare. Okay, people with this blood group are very rare. That's it. It is called rare blood group. But in your class, you get at least 2 to 3 people. You get teacher asks, hey, all A positive. Raise up your hands. 10 people will stand up. A B positive. 10 will stand up. Hey, O negative. You will be searching. Oh, it's not there. Just go to the next class. You can't even find it. In every class, you can't find one A negative or O negative blood group. Can you understand? So that is very rare. Okay? In your family also you can do an analysis. What the gene is dominant.
I don't know, we went to an accident,
Hmm.
he's on third loss.
Hmm.
What if the person is different that gives him blood? What happens?
Still listen. In World War II. Okay, still listen. In World War II. Most of the medical discoveries, for soldiers especially, okay, in those days, okay. So, something called if the soldier is injured, blood transfusions can be done. But whenever they do the blood transfusion, some people are with little injury they die, but with major injury also they are safe. And finally they came to know that actually, okay, blood is not matching, and then they tried, okay, get blood from Anil, get blood from few whatever blood from Ali, just mix it in the lab. Okay, both are matching actually. Then Hari can give blood to Anil, matching. Okay, so no antigen antibody reactions. If in B blood group you mix it with A antigen and B antigen interacts with each other, clumping blood clots. If I am having A blood group, you are having B blood group. Okay, if you give me blood, my blood clots, antigen antibody reaction. If my blood clots, heart attack, I will die. Not, yeah, because the O positive in the sense, no antigens are present. If antigens are absent, how it is going to react? If O, only O negative present. Rhesus antigen can cause clotting. A antigen can cause clotting. B antigen can cause clotting. Okay?
Sir,
If he is having AB blood group, he can get A antigen, A blood group. He can get B. He can get O. They are universal acceptor. They are called what? Universal acceptor. Okay, everybody's blood is they can accept. All antigens are present, all antibodies are present in their blood. So no problem for them. They get all blood groups. They are universal acceptor. O positive people, actually, O people, they can be what? Donor in simple. So they can no antigens basically present in second give actually. Okay, so everybody can get their blood, especially the positive, not negative people, but positive. Okay, this is positive, it should be positive. O negative, only O negative. Don't involve in that. But A positive can get O positive blood. B positive patient can get O positive blood. AB positive can get O. So they are what? O positive. People are called what? Universal donor. They can give blood to anybody. Because they do not have antibody on their surface and no cross reaction occurs. Can you understand? But this is rare, but only they can get blood from them. Okay? Similarly, the A negative, very rare. So they cannot get positive blood. Okay? O negative people can get, O negative people also can donate to everybody else. But they don't give actually, they don't allow because it's very precious. O negative blood is so precious. Okay, rare. Not everybody can get it. Okay, only the people request the same blood, then they receive it actually.
sir, can A cross the blood can, is it get worse?
No, no. No. Okay, because in A blood group, B antibodies are present. In A blood group, B antibodies are present. If B antigen comes, antigen antibody reaction occurs, blood clots. He cannot get it actually. Only from A B positive. O positive also can be. Okay, you can give two O positive people. Okay. Anyway, so DNA copying today about what we discussed. Main thing is about mechanism in sexual reproduction or any reproduction which is DNA copying mechanism. Okay. Okay. Before finishing the class, some simple questions. Okay. What's the elongation process? Size as well as contents increases. Okay. Elongation process. What is the next process followed by? DNA replication. Replication means what? That's the DNA copying mechanism. One DNA produces its own copy. One DNA produces its own copy. Okay, that is DNA copying mechanism. It should be followed by nuclear division. Division of the nuclear. Also called, what is the technical term? Karyokinesis. What is the next process? Cytokinesis. Means this division of the cytoplasm results in formation of two daughter cells. Okay, see why it is called daughter cells, why it is not called son cells. Yeah, common term, especially like humans. See, all science is based on anthropocentric. See, listen. See, we study biology based on what? Anthropocentric biology. Biology is anthropocentric biology. What is called anthropocentric biology? Human view. Human view. We call it as what? Anthropocentric. In humans, female only produces offspring. Appa and son cannot produce offspring. In the sense, only from mama and daughters, another progeny is formed. That's why they are called what? Daughter cells, not son cells. Okay? Fine, good. Anyway, so what is variation?
The progeny differs.
Differs. The degree by which the percentage by which progeny differs from the parents. Okay, that is variation. Yes, Kaushik, are you there? No. Okay, accumulation of variation results in speciation. Okay, in what percentage my DNA matches with other DNA? All humans, 99.9%. Okay, fine. So keep these things in mind. Simple questions we'll discuss in the next class. And then we will go to the alternate questions. Yes.
Define the terms in the mistake and the leads can mind that on this leads is it going to happen?
Because your blood is not going to enter into that bloodstream. It cannot enter. Lot of filters are there. Okay, just like that cell is bigger. Okay, it cannot enter actually. But if your blood is having bacteria, the bacteria can enter into another body. The bacteria enters, they can easily pass because they are small, smaller than your cell. So they can enter. So your blood cell cannot enter, but your blood cell which carries the bacteria or virus can enter into one. Okay. Good. So one month. Yeah, means sir, if a drop of blood uses, will it be affected? Is this your question? No big difference because diluted. Your antigen can be destructed actually. There little dose of antigen enters, it get destructed. No, it cannot find much company. One antigen molecule can get one antibody molecule. At least one can, like a waste, it can be excreted. Okay, but if it is more only, lot of blood can fall. Okay, fine. Good. Thank you. Take care.